trying not to overlap too much with what Chris did. So the microbiome contains over 100 times more bacterial genes in your body than there are human genes, which means that it is an amazingly complex ecosystem living inside your body. There's over 2 factor difference of 10 actual bacterial cells in your gut than there are human cells in your body. So we're talking about massive amounts of diversity in a relatively small area, which makes it a very complicated system to work with. We used to think that the human gut microbiota was just there to regulate metabolism, or what energy we could bring in from certain foods. How it works is you eat some foods, and then since bacteria are masters of metabolism, they're able to use many different types of metabolism. They would break down anything that our body couldn't break down, would take those extra metabolites, and we would then use those in our body for energy sources. But as time has gone on, we've realized that immunity is now heavily modulated by the human gut microbiota. So in 2006, Bruce Lake from Cornell showed that obesity-induced mice, like these here, had a structural shift in the microbiota. Specifically, there were large phylum of microbes that were found in the obese that were in different proportions in the lean mice. So obesity is induced here by a gene knockout of leptin. Leptin is a hormone that induces satiety. So without it, mice eat, they get fat. And there's normally a high-fat diet here that's also added. So what we saw were these large shifts in gut microbiota, which was our first indication that it's playing some type of role in obesity. Next, Peter Turnbuck, also working with Rugley and Gordon at University of Washington, showed that obesity could be induced in mice by transplanting fecal samples. So taking fecal samples from a fat mouse, putting them inside of a lean mouse, and basically seeding the lean mouse with the gut microbiota of the fat mouse. And the lean mouse becomes fat, and opposite, the fat mouse becomes lean when receiving samples from the lean mouse. Once again, heavily showing that the microbiota is somehow aiding in obesity. So, what's going on with inflammation here? Why do we keep talking about it? How is it related to obesity? And this is all because of a particle called LPS, lipopolysaccharide, more commonly known as endotoxin. So, all gram-negative bacteria, as anyone with basic microbiology knows, has an extra cell wall on the outside that is made up of this LPS, lipopolysaccharide, or endotoxin. When the cells break down, this is excreted into the system, into our gut, and it moves through the gut and attacks macrophages. Now, Buehler and Hoffman in 1998 discovered that this LPS binds to the TLR4 receptor, toll-like 4 receptor. They received the Nobel Prize for this in 2011. So, just a hint at how gut microbiota is changing global science. So, it binds to this TLR4 and initiates a metabolic pathway that's studied by Gokhan and Mitchell at the School of Public Health at Harvard. And what it does is it produces this NF-kappa B. NF-kappa B upregulates producing of pro-inflammatory cytokines, most prominently TNF-alpha and interleukin-6. Now, TNF-alpha and interleukin-6 then go through this whole pathway and activate and upregulate J and K, which dephosphorylates this IRS1. And if you come over here, you see that IRS1 is part of the pathway that brings glucose into the cells. So, when we phosphorylate it, glucose cannot be transported into the cells by insulin. Now, as you guys know, diabetes is basically the inability, because of insulin resistance, to bring glucose into the cells. And what we're seeing is this inflammation is targeting that pathway. So, in 2007, Kami introduced the first evidence that high levels of endotoxin in the bloodstream and in the serum stimulate obesity and insulin resistance in a human sample rather than just these nice molecular targets that a lot of people like to work. So, we now have inflammation, obesity, and the microbiota. We wanted to see exactly which endotoxin producers were available. So, we took a patient. We had a 16-year-old type 2 diabetic patient who had a very high BMI of 42.93. And using previous studies done by our graduate student Ray Ray, we found that we took fecal samples for a couple days, for 75 days, and we found that Enterobacteriaceae, which is an endotoxin producer, was very prominent in the sample. This is qT-PCR data, which is what Chris did. And we see high levels of Enterobacteriaceae. Also, we performed 454 sequencing, and we see this large chunk over here, which is Enterobacter. These two are Bacteroidetes and Firmicutes. They are the most prominent microbiota, microbes in the microbiota. And basically we've realized since they don't decrease or increase very much throughout time, and they're quite stable, that they're not the ones producing the endotoxin that's affecting the system. But when our patient went on therapy of both diet and exercise, we found that we decreased Enterobacter. So we thought that would be a prominent group to start and look at for endotoxin producers. So, I'm fast forward.
Okay, I'll introduce the method and the results we get in this experiment. First, let's have a look at the overview of our project. First, we activate sample from the fecal sample of our patient, and then we do phase dilution to capture bacteria in aerobic and anaerobic. And then we do cage. I will explain this next. And we do cage to isolate positive strains, which is Enterobacteriaceae. And then we do other and Eric. This to molecular typing to subgroup our strength, and then we sequencing using RNA of representative strength. First, for aerobic culture, we get 6 type of morphology. Actually, type A and type B is the majority strand of colonies, and for C to F, there are just several colonies. And for A and B, amount for half. And very easy identified where A is a large one and B is a tiny one. For anaerobic, we get two kinds of morphology. One is dark and one is light, and also is very tough. And this is for I think recording experiments of our project. It is colony institute hybridization. We use it to isolate Enterobacteriaceae. We call it cage. So when you see this simple illustration, first we transfer colonies from original phase to nitrocellular membrane, and then we lysis the bacteria and bind DNA to the membrane, and then we use probe to hybridize, just like similar to sound blocking. The primary, the probe we use is specific for antibacterial state. It's ENT 183. And then we, after hybridization, we use immunological detection to show the positive strength because our probe is labeled by PIG. And we use NPT PCID color solution to activate color because we don't have a machine for fluorescence, so we use this method. And I'll show you the results. This is the final result of the case. You can see here is this light color is the positive control because it's not in the original phase. We just put it into to this membrane, and for this is all from the original phase colonies. This dark purple and the colony shaped patch shows the position of positive strength. And we compare this membrane to this phase, and we can get the antibacterial acid. And we calculate, and we do molecular typing to identify those strengths. For other, it's a method we amplify 16 siRNA and digest it with restrictive nuclease. We use two enzymes, CSP, CS1, and P1. This arrow picture shows the result of P1, and we can see left band, different band patterns. And the lower picture shows the result of ERIC. ERIC is similar to random PCR. It's used to repeat the repetitive the genetic consensus fragments of DNA. And this is the final use. And actually, we do the over 170 strands of the positive constraint and succeed for all other in the area is about 118 strands. And we conclude all kinds of other and combine them together in one job. But in this really is not very good. And we have 4 pendants for CSP 61 and 4.1, and we have different pendants have combinations. For Eric is really a tough job on many gels, and we conclude 4 pendants, and it's a majority pendant. It's between gels and I want to share a very funny story about how we pick out these 4 pendants. You know, for Eric, you can see this is in one drawing. This is a fine gel so it may be easier. For this is similar, and this is different. But for different gel, it's really tough. You see the band position, and then and each band intensity. We find very tough and boring. And now I think the music can save sound. And you see, for pen 2, we can say boom, 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 boom. So it's easy to remember to see pendants between gels. So it really reduces many works. And last, we sequence 16 sRNA. And then we pick 16 representative samples. And for this tree is from rocket culture. And these samples cover nearly all the canton of culture, and the majority canton of poetic. And this is the other kind of poetic. And after we get the sequence, we do cluster in SPI, and we find the many is 100%, and we pick out the highest score, and this should be the closest strand to our samples. And we can see for this cluster, nearest stem, and that shows many of them is coli and Shigella, and then we match it in the screen gene data, and we get a similar result. For this class, here's the same, and here's the same Shigella. This is the genetic tree we get by doing genes, and like last slide, this strand, I think we will take it from this tree close to the neighbor from sample isolate, and here's not, and here's two. And there's from our, as also E. coli and the Shigella.
So I'm going to quickly wrap up with the discussion of fusions. We're running low on time. So what we managed to do this summer was successfully establish KISH in the lab and prove its effectiveness in isolating Enterobacteriaceae. This is really important because what we've realized now is that from a cultured bacterium, from a culture or fecal sample, we're basically able to isolate any type of family that has a probe and directly take them, move them out for positive isolation sequencing, which is a big time saver. The test takes 3 days, but it's better than culturing for 5 different times for 5 different days. We also realized that a combination of Arthur and Eric helped identify different strains of Enterobacteriaceae. This helps to pool our strains together and send them out for sequencing, so we're both not wasting money and time. And finally, and most importantly, we're able to isolate 16 strains of endotoxin producers from a morbidly obese patient. So these strains were cultured both aerobically and anaerobically, but in very unselected media and very unspecialized media, which means that there are probably very, very high populations of these specific endotoxin producers in the gut. And that means that these specific strains are probably very critical to what we think is endotoxin producers activity in the gut. To further do work here, we've realized that there are very close phylogenetic similarities between Shigella and E. coli, which means that we need to do further molecular biology in order to tell the difference between them, not just our blast matching and our sequencing. And finally, we need to get a chance to quantify and identify the type of LPS that are in these endotoxin producers. LPS has very long chains on the end called the O antigen, which tends to differentiate it between different species and different strains. So it could be that these strains produce a specific type of LPS that is more likely to bind to TLR4 or has more potent effects in the body. But we won't know that until some further analysis. So just to wrap up, we want to thank everyone for all of their help, especially Alana and Meeting for setting this project up, setting this program up, Professor Pong, Gregory and Pena, and all the administrative staff that helped make this thing happen. So thank you, everyone. Any questions? And they need to be quick. We're running out of time. The pressure's on. No.
How many how many times?
How many times? That is a question that many, many people are trying to answer. At the moment, we're looking at probably 5,000 plus species, but that does not include the potentially 200 or 300 strains or 300, 400 strains for every one of those species. So we don't really know any difference between person to person. I mean, I do not have the same microbiota as anyone in this room, both because of who I am and what I eat and what I do and where I live. But mine stays pretty constant, while everyone stays pretty constant. So normally you can tell the difference between one person and another, but the difference is larger between people than it is for one person.
During the reason that you will eat the outbreak, your second, so some bacteria infection may on the reason
Yep.
the treatment.
Several bacteria have been linked to mental diseases, and now one of the big deals is looking at some of the gut microbiota and seeing how maybe some of this inflammation pathway causes these inflammatory nerve diseases that we're seeing. So, very interesting. Thank you.
Hello everybody, I'm Dan, and also your newest to get us to make the presentation, tell us about our work during this program. My name is Zhang Xiaolong, student in I'm going to be a senior in Castillo, and this is Derek.
I'm David Kingpon, and I'm a rising sophomore at Harvard University. The research project we did is called the General High Throughput Screening Method for the Directed Evolution of Enzymes. And we're just going to briefly how we're going to start. I'll be talking more about results, and then we're going to combine and wrap up for future research.
In this presentation, we have 5 parts. The first part is introduction. So I'm going to make some brief introduction on the things we do in this project. Here it is. As everybody knows, the enzymes are very efficient catalysts for to make single or complex molecules, and in the cell, it is very attractive for industry to put it into commercial use. But there is a big problem that sometimes the enzyme isolated from the nature can work very well in industrial solution. So here is a way to change its property, to improve its property. The directed enzyme evolution has proved to be a helpful tool for improving a range of enzyme properties through consecutive round diversification and deletion. And now modern technology has allowed us to get tens and hundreds of thousands or even up to millions of mutants very quickly. But there another big problem comes. How can we select the useful one or the efficient one? We want gap. So in these recent years, there is another method to select and screening the mutants. That is the fluorescence, active cell sorting, FACS. It has been a very powerful tool to select the mutants, and it is very efficient because in one day we can even get greater than 10 to 8 mutants per day. So like in this picture, we get the gene library and we transform into the cell, and we can make the expression of the gene to get to the enzyme, and then we can make the enzyme, make the cell into the water and oil system, and then we can add the substrate. So it has been special design, and it contains the fluorescence after the reaction with the enzyme, we can get fluorescent product, and then it can be tested with FACS. And then in this picture, it shows how the FACS works. And after we get the water and oil emulsion system, then we use the machine to test it. We use the excitation laser test it. And we can get the stronger and or weaker fluorescence cells, and we can separate them from each other. And it can be charged, and the stronger one is here, and the weaker one is here. So using this method is very quick and very efficient to get efficient mutant. But then we face another problem. Because in this picture, you can clearly see sometimes in the emulsion, sometimes there are two cells. This will be a false positive one. Because if you want to get a very efficient mutant, you want to get that is only get apple. You want to get something that is greater than 1. And this one, each of this one is 0.5, 0.5. Meaning 2 add together will just be 1. So it is false positive. So what we want to do is just to delete this false positive. So we need to construct a string with another fluorescence label gene, the MCLE marker gene, and we need to gene locate to the cell. And then Dale is going to talk something about the mass. Oh, sorry. I'm going to class. We're going to talk something about the preparation before we do the main experiment. The preparation, we need to get the efficient in the cells, and we need to select the suitable promoter sequence for the fluorescence. We have made 4 strings of covalent cells and contain chemical covalent cells and electrical covalent cells. The chemical covalent cells top 10, this one, P109, and we have tested whether it works well or not. So, and top 10 beautiful plasmid attraction, and GM109, and is used for getting transformers, carrying red plasmid, hyperplasmid, to help the genome, to help the genome. And this picture shows we try to find the suitable promoter. In this picture, we have transformed 9 different plasmids into top 10, and each alpha. We want to get the strongest fluorescence. So we just use our naked eyes to distinguish them, and we get this one, PZS.28, P-type and cherry. And in the next in future experiment, we just use this plasmid for the program.
So I'll briefly go over some of the methods that went into this project, and giving a review, there were 3 main components. The first was to construct a new plasmid, and going along with that would be to engineer a strain carrying the helper plasmid in this genome process, and then finally to combine those 2 into the newly engineered strain that we want as our final product. And so this schematic here really shows that the PKD3 has the FRT and a fluorochemical resistance component, which when added to the promoter sequence of M-cherry, which adds autofluorescence to the strain we're looking at. When combining those, that allows for selection for the when we're creating our final strain. And I'll get into this more later. I just wanted to bring special notice to one of the papers that published in 2000 that was really the basis for a lot of our methods and protocols, especially in creating this target strain. You can see just up here that it's been cited in over 1,000 publications because of the way that it allows you to do one-second activation. If you grow the strain at different temperatures, it then allows you to either have that resistance gene work and operate or not. And so it makes the process for us and this genetic engineering in general much easier and faster. And so the main methods, because we have more of a qualitative aspect to our results rather than quantitative, this is just a textual overview, and I'll be showing really what we were doing through each of the gel pictures that really shows each step of the process and should really confirm each step. But basically, we wanted to take the insert fragment, amplify it through PCR, digest it with the AAT2 and JO1 enzymes. Then we would get that vector plasmid, transform it, extract it, ligate it, and then transform it into top 10 to confirm it, and then harvest the new constructed plasmid. Then moving into the second step of transforming PKD46 into JM109, that was to make sure that it had the helper plasmid within it, and then we also made electrocompetent cells in order to finally complete the third step, which was the gene knock-in and making the final strain that we were looking at. So what we accomplished through this summer's project, looking here, I won't zoom in too much just so for the sake of time, but each of the gels here shows a different part of the process. So the first part is just extracting the plasmid and then purifying it after digesting it up here. We then purified the PZF*28M cherry. This is the promoter, which we transformed into the strain of interest. We then went over to, we went down, and we, as part of the second step, we digested the insert fragment, which we had amplified through PCR. And we combined those two in terms of showing them on one gel right here, the ligation, as well as the purification of the plasmid. We were able to successfully show get colonies from our transformation into top 10. So we did colony PCR to confirm that it was indeed the right length and the right size. We then used this to get this gel down here, which again shows further confirmation of our work that our transformation is successful. We do have the correctly engineered strain, and we were able to transform it into the top 10 competent cells. And so the final step of our procedure, as you can see, it would be to take the insert fragment that we have, which has the FRT and the resistance strain, as well as the P-type cherry promoter, and putting that into the JM109 competent cells that we already added the PKD46 helper plasmid into. So lots of different transformations going on all in one place, but in the end it all comes together into forming the final strain that we desire. And so you can see the most recent results from just last week, the end of Friday and this weekend, is successful transformation of the colonies on this plate, and doing colony PCR and extracting the plasmid to verify that it is indeed the right size and shape.
Okay, because last week we had just added the efficient common cell GM109. So just this week, yesterday, I've just transformed the fragment into GM109. And something grows on the plate, but we have still to make some experiments to make sure that it's the right one that we want to get. So, and if we get the target strain, we can use ACS to test whether it really works or not. So, and other side, we can find some places for improvement of the strain. And then in this program, we just use the genetic ways to construct a new strain for the after ACS to make some improvement of this method of that system. And we have almost get the strain we want, which can be very important and high throughput screening of enzyme library.
So just to wrap up, I know we spoke very rapidly at times and it may not have been understandable with all the transformations and the gels taking place. But in the end, the final implication of our research, and as this puts us towards future research as well, is that the current method of screening high numbers of mutant libraries is that it takes to do it by 96 well plate, it would take several hours, several years for someone to screen a library of 10 to the 8, 10 to the 90 mutants. And by engineering the strain, we're able to have a strain that autofluoresces that can be combined with current technologies already to make it much faster and to reduce the number of false positives. Some statistics show that about 50% of what you think might be good data when you first are looking at mutants is actually false positives. And so this helps eliminate that possibility, saves a lot of time for the researcher in terms of genetic engineering work, and makes things a lot faster. And so in terms of working with other people beyond just our time here at Georgetown University, is combining our experience with others in the lab who are using the lab-on-the-chip technology to create these water and oil and water emulsions, which we spoke about briefly earlier, and to also just, once we complete the strain, to transform in the final gene of interest or enzyme that we're looking at, to do error-prone PCR, mutagenesis, and to test a very large mutation library for whatever desired effect we want. And so finally, we just want to thank, and use the same picture as Selena in thanking both the lab and everyone who supported us, but also draw special attention and note to our advisors in the lab, our grad students, Hong Chen and Mong Chung, and then Dr. Chen and Dr. Feng, as well as Dr. Beal and Dr. Zhao, and Grace for all of their support. So we're very appreciative. Thank you.
We have time for a couple of questions. This one was shorter. No questions?
Actually, the knockout procedure.
Yes.
So can we use that same protocol packaging some other genes that are interested just to specifically knock out the gene? Are you using the same procedure that you use?
Yes, although I'm a little bit confused by your terminology because we were doing we were adding a gene in, not knocking out.
So
It was a gene.
knocking one gene.
Yes.
Not at a specific place.
Yeah, correct. We were looking for a specific gene. Well, thank you, and we'd be happy to answer questions otherwise after all the presentations are over.
Hi everyone. Let's give another round of applause. Everybody worked so hard. So to finish up, we actually, some of the Harvard kids have some things for you guys as a token of our appreciation. I have the picture of the guys, but so actually, if you would like to come up here, we have something to give you.
Okay.
This is a Harvard notebook.
Oh, okay.
So you can take it to your meetings.
Yes,
to
yes.
conferences.
Thank you. Okay. Thank you.
Yeah. And so for everybody else, we got you some gifts as well, but you know, there's so many people. But your undergrads are really working with you. But thank you guys so much for an amazing summer. And it was a lot of fun to work with everybody. But it's not over yet. Reports are still due, so you know, don't relax just yet. But you know, it looks like everybody's off to a good start, and we're really excited to see how the reports are going to go deeper and get more into the science and the discussions. So, thank you.
Can I just say a few words?
Yes.
Okay. Thank you. And I really enjoyed every bit of this symposium. And thank you so much. It's really a wonderful experience. And you know, I didn't bring my cell phone to the meeting. This is very rare. Okay. So I realized that sky is not falling if you don't answer your phone for a few hours. Okay. And then you can have time to really enjoy the good science and the good spirit and of our very fresh future scientists. Okay. And so I think we should thank first all the Harvard and Georgetown students for your hard work and for your dedication And I know it's a wonderful experience for every one of you, and including some bumping experience on the road. And but you all survived, right? And through the hard working, and then you get establish a new friendship, and so this is really a wonderful experience. And I also would like to thank our supporting staff, Grace, and Chen Feng, and Hanjin, and Wen Chun, and Bai Liang, and everyone who have helped. And also I would like to thank all the professors in the hosting lab and their staff and their graduate students for making this happen. And I hope this is the beginning of our collaboration, and we'll continue this as a tradition. Okay, thank you very much, and enjoy your summer. Okay, bye-bye. So, if you don't want to leave, you can stay here.
Okay, that was great.
No, but I need help.
Yes, yes, I heard your name. You only have two, right? Yeah, I only have two guys. You're giving up. Oh, I have